All right, so I'm Brian Woodfield. I'm sitting alone in my department in my office at BYU. I'm a professor of chemistry at Brigham Young University, so I created these labs, and we've been working on these labs for 20 years. They've gone through a lot of different iterations. They were originally available for almost 15 years by Pearson Education. And but recently we took them back from Pearson and have they were developed at BYU, and we've been updating them, and we have new versions that are now available through Beyond Labs. And so that's how you've reached out to us. And so we have labs for general chemistry, organic, physics, and biology. So I'm just going to talk and give a demo about how it all kind of works. So kind of bear with me a little bit, that I know you want to see the labs in action, but let me spend some time about how you get to the labs, and where the worksheets and lab activities are, and some other maintenance questions that students will always ask first. So I'll spend maybe 5 minutes on that. And then I'll start doing some demos of the three labs and kind of switch between them, so that the biology, the organic doesn't feel left out for 20 minutes or something. So I kind of go between them. They all kind of work the same. And so I'm just going to start sharing my screen with you now. So I'm going to share that. And I'm not going to be able to read your text messages, but if you got questions, just text them to Heather, and Heather will kind of know where I'm going, and she'll interrupt me as necessary to answer questions. So feel free to ask them.
Or ask them directly to the group. That's fine if they're in the general strand.
Yeah,
text
to her or to the general group, and she'll check them. So, all right. So this is the client. So you go to Beyond Labs and you download this client. The students would. It's a small download. It's free, and so you download it, install it, and it works on Macintosh and Windows. And we do have Android versions for Chromebooks, but those are really designed for kind of high school applications. So you have these tabs across the top where the labs are. So these are all installed except for physical science. So if you had this running and you wanted to go do physical science, you would click download, and it would download the app, install it, and now it's available for you to run from the client. We have lab activities we call worksheets, in the form of worksheets. So we have each of the different products have their set. So here's chemistry, here's a high school version, here's the higher ed, and then they're divided into these different subjects. And somebody mentioned spectra, so here's atomic emission spectra, for example, a worksheet on that. And so these are PDF files. So for example, if you wanted to do Rutherford, you'd click on that. It opens it as a PDF file. And these are the lab activities, so they have some background information. And then they have procedures, and explain everything that students should do with assessments. And we'll get to later at the end about different ways that instructors, professors, teachers use these. But for right now, this is kind of how it works. This is a drop-in solution. You can say, hey, students, go do the Rutherford backscattering experiment. And that would be all you have to do. Students would follow those directions, go to the lab and use it. I'll show you how that works in a minute. But these are all of our content. If you message Heather, she can send you, we have a list of all these contents in a nice, easy file to read. We also have installation instructions for you to use. So you have to activate the product for it to work. It comes with a free 2-day trial period. And but you can purchase codes. We can give you trial codes if you need longer as the instructors. Typically, students will purchase it for $25. They get a code, they install it, or an institution will purchase. And then you tell us how many codes. We work out a price for those X number of codes, and we'll send you a single activation code that works X number of times. You put it in this box, click submit. Now the product is activated until some date. I don't think we have any high school teachers, but this is how you change the level. It doesn't change the labs. It just tells you how to change what worksheets you're going to be using in the lab. So licenses, worksheets, then you come back to the labs, and that's how it works. So this is just really the hub, but this is where the labs go. So I'm going to start with general chemistry and first, and do a few things there, and just touch base for all three, and then kind of come back based on your input on which ones you want to focus on. So let's say, for example, they wanted to do Rutherford like I just showed. So a student would have that PDF up. They'd come over here, they'd start chemistry, and chemistry starts, and this is the landing page for chemistry. And so you would see these different lab benches. So all the labs are divided into lab benches. Each lab bench does a particular class of experiments, but most of them do a lot of different kinds of things within a particular kind of experiment. So for example, titrations would do, obviously titrations, but it does oxidation reduction, it does pH meters, it does, so you can talk about buffers, you can talk about strong electrolytes, weak electrolytes. It has a conductivity meter there. So lots of different things you can do within each one. So those same worksheets that you saw in the client are all divided in the same way here. So what you would see here is you see the same list of titles for the worksheets as you saw there. You can always click on any of these to go into the lab, and it's just going to be like opening a door and going into a room with this lab bench full of things to do. But you could also use the worksheet. You would click this. So I'm going to, this is one of my favorite experiments. So I just clicked it. Notice that this is the quantum lab. It's already preset. So the labs come with the ability to preset themselves into certain configurations that match the worksheet activities. And so the student would have the worksheet available either as a printed document or as a or online. They would follow the instructions and then go through and do this experiment. So just to give you an idea, this stockrooms have, these labs have stock rooms. So here's the PChem stock room, the quantum stock room with all this different kinds of materials, and has clipboards, which we'll show you how to use in a minute. But here's Rutherford. So it's got an alpha particle source, it's got gold foil, and it's got a phosphor screen. So I can turn on my phosphor screen. I'm going to move my bench over here out of the way. And so there's the Rutherford experiment, shooting alpha particles through a gold foil. But the key thing here is that it's open-ended. You know, what happens if I shut my shutter and I keep them from coming out? Then I get no signal. Open it up. What happens if I move my gold foil over here? I get a small spot. So I put gold over here, I get a fat spot. Why? Those are questions we're going to ask them. What happens if I move my phosphor screen over here to the side? No fat spot, but I still see alpha particles being scattered at a wide angle. What happens if I move it over here? Looks like I don't have anything, but I'm getting a few. So I can click here and persist, save where these things go. I can also click record and record this in my lab book if I wanted to. And so this is how the Rutherford experiment kind of works. So I'm going to unpersist this. And so now we just done the Rutherford experiment, but it's completely open-ended in that a student could come over here and say, well, what happens if I change my metals? So I'm going to go to my, I'm going to go over here, I'm going to return my gold and click on my box of metal foils, and I have all of these different options I can use. So what happens if I go use, say, magnesium? And then I can return to the lab, put in magnesium, and I see a little bit bigger spot, but I don't see nearly the amount of scattering that I got with gold. So lots of cool things that we may not have a lab activity that we discuss really, but all of these options are available. So, so that's just an example. And you can build these experiments. So you can take any source with detectors, with sample holders, and with samples and modifiers, and do these any experiment you want. We also have these clipboards that preset ones. So here's Thomson experiment, for example. So I got electrons coming out at 100 eV, going through electric fields and magnetic fields and hitting my phosphorus grain, turn on my grid, and now I'm going to apply an electric field here. So I bend my beam, and now I'm going to go add a magnetic field and move my beam back. And then from that, they can calculate charge to mass ratio. So that's just an experiment. Millikan oil drop. So here's oil mist. These are all preset, but they can build them. I can apply electric field and move my drops. So I got random size drops with random number of particles. You know, somebody mentioned some spectroscopy here. So I got 2-slit diffraction with light. I can change my slit spacing. I can change my wavelength and see how my diffraction pattern changes. I can come over here and change how many photons are coming through. I can click persist and teach them about Heisenberg's uncertainty principle using 2-slit diffraction and have them actually experience and build up the diffraction pattern over time. Absorption of HCl, maybe that's not a freshman chemistry thing, but we have all of these different experiments. You can zoom in, see these, and see the spectra and talk about it, or you can do photo emission of CO2, for example. And the visible spectrum here. So all of these kind of things that you could do in quantum. So I kind of showed you a lot in one of these labs. For general chemistry, we have, you know, so we generally have these labs kind of divide themselves into two kinds. We have simulations of labs that you would actually do in a real lab for your students. Quantum mechanics happens to be an example of a lab that maybe your P-Chem students might do it, but your general chemistry students wouldn't. So, but they wouldn't have access to because the equipment's not safe, or it's dangerous, or it's expensive, or just takes forever to do it. But here that you can do it. But we also have, you know, simulations of a lab that you would do. So here's inorganic qual. So so here they would grab a test tube. And now I'm going to show them a real picture of what's in that test tube. All right. And then you have these metal cations, add them any order, any combination. So I've added cobalt, chromium, copper, and this is a known. So now they can do the chemistry, and now they can do these lab manipulations. So here's a flame test, for example, and it shows them the green from copper. And here's a flame test with a cobalt filter. And then decant, divide, heat it up, you apply heat to it, and then you have pH of the solution, pH of the vapor, smell, stir it up, and then you have these 11 reagents that you can add. Any order, any combination. So you have 25 cations plus water, and these 11 reagents with heat and all these things, and so you have about 10 to the 26 real outcomes. So all of these outcomes were measured. Obviously, we didn't do 10 to the 26. That would take a long time. But we devised an algorithm based on real data. So we took the chemistry of all of these and measured all of that chemistry. So if I add pH 10, look what happens. What if I go to pH 4? Dissolve the precipitates. Go to pH 10, I get precipitates. I'm going to centrifuge it. There I force it to the bottom. What if I go to sodium hydroxide? Well, now I have chromium, which is amphoteric, is in solution, but I got these cobalt and copper. So now it's centrifuged, and I can do a decant and separate that product. Now here's my chromium. I could switch them oops, missed. I could switch it and do some chemistry on the chromium by itself if I wanted to. Um, and then we can do some chemistry here. So here's ammonia. So I've added ammonia, centrifuge that. So my copper is in solution, my cobalt isn't, but if I go to pH 10, they both are. If I go back to NaOH, then I get the one in solution. So I centrifuge it, decant. Now I've done a separation. You can do unknowns, okay? So you can come to the clipboard here and pick different kinds of unknowns for you to grade that get reported to the lab book. You can also create these practice unknowns. So I can pick a minimum of 0 and maximum of 3 and pick my cobalt, chromium, copper, and now I can create an unknown here. And so I can grab that unknown, take it to my rack, move this guy off, and put this on my rack. And now I have an unknown. And I can go through the tests, and then I go to my lab book here. See that I've saved that phosphorus grain and do a report. I know I have chromium at least, submit that, and I got lucky and got them all right. So lots of different ways, of using this. This isn't just a qual scheme. This is a great way to teach your chemistry, to combine acid-base chemistry with redox chemistry, with complexation chemistry and precipitation chemistry, and to reinforce all the chemistry that we teach in a general chemistry class. So just really quick, because I know I promised to go to the other labs, titrations, just showing you what it is. You have pipettes, you have beakers, a burette, you have pH meter, conductivity meter, balances. You have a stockroom full of acids and bases, and you can do all these different kinds of titration. So here's just a really quick example of a titration with a unknown acetic acid concentration with sodium hydroxide. It's all set up. This is a preset, all set up, ready to go, but you can kind of see how we can have them go do a titration here. And there's real errors introduced here. These burettes are not perfect. They don't deliver perfect volumes. The balance isn't perfect. There's buoyancy corrections. And so students are going to get real data. I can save this data to a lab book, for example, be saving it while I go through. We have indicators. And so we'll go through this endpoint here and click stop and open my lab book. And here's my data that I can now copy this data and paste it for them to analyze outside the program. We can save these lab books externally, send them to you, or open them back up, save them for themselves, so they can come back with the same lab book. Calorimetry, just to kind of show you the lab, same thing. It's got beakers, and in this stock room you have coffee cups and dewars and a bomb calorimeter, and you have drawers full of all these different metals. So you have 64 different metals for which you can measure heat capacities. You got organics for combustion, salts, reactions that you can measure, clipboard full of experiments for heats of solution, boiling point elevation, and so on. So that's just kind of what you can do in calorimetry. So I've been going really fast, so any questions on general before we kind of move out to the other ones?
Could you show how the lab book works and how this integrates with an LMS, so how professors could be accepting assignments from students?
All right, good question. So if you go to these worksheets, for example, and go back here to acid base, here's a acid base titration. This is just a worksheet where they could go fill out their data. And so in this unknown, they would just report what their concentration is in this unknown, and give this as a piece of paper. And we would give you the solution keys to all of these worksheets, and you could just grade these by hand. So this is a really easy way. Here's a worksheet. hand it in either on a piece of paper, which isn't going to work right now, or they take pictures, create a PDF, upload it into your LMS. Or sometimes people just say, email me the answers or give me a simple document in the LMS with the answers on it. That's one easy way of doing it. The other way in the lab book itself is, so let me go back actually to titration, for example, as an unknown, is when I pick this unknown here, for example, weak acid, strong base, it gave me unknown 11. So that tells you in the key, unknown 11 has X concentration. So that's one way of doing it. The other way is you can go down here to these unknowns and say, hey, I'm going to give you a KHP unknown, 90 to 100%. It's going to be somewhere in there, and you're going to return, and it's going to give me KHP unknown, and I have no idea what it is, right? So I would come over here, and I would say, okay, I'm going to go with this known concentration of NaOH, return to the lab, and I'm going to go do this experiment. So I'd have to come over here, go to my balance, put on my weight paper, tear it out, open my lid, you know, add some amount, and you know, oops, I forgot my beaker, so I got to get a beaker over here. I'm not going to do it all, but you can see how they would actually go do this. And then when they're all done, you click your lab book, and you notice here that it has report, and they would just input their answer. So 95.34, click submit, and you get the real answer here. And this gets saved as a link in their lab book. Then what they would do is go to File and save this lab book on the desktop, and it's going to get saved as a lab book as a file name, and it's going to get saved. And then what they could do is come back over here on File and open it. But what you do as a teacher is you would just say, hey, upload this electronic lab book file to your LMS, and then the TAs, or I usually have my TAs, download all of these in a folder, and then they go through and open them one by one and look and grade them. So Inorganic works the same way. Calorimetry has some unknowns, works the same way as well. Does that answer it?
That was great. You're doing great.
All right. Let's do biology so to make sure that we don't feel like the whole world is chemistry, and focus on chemistry and organic. So same idea, same landing page. We have these different lab benches, and I can't show all of them. And we have these different groupings and different experiments that we've done here. So I've noticed here that this is the high school version. I'm going to kind of show you. See here it says high school. I'm just going to show you how it works. Is I'd come over here and just say, I want the higher ed version. Come back to Labs. It says higher ed. I'm going to start it. And the labs don't change. What happens is the titles that go with the worksheets and the presets change, so they match the different levels that go on here. And so same idea here. So obviously, I'm not a biologist. I'm a chemist. And I had two co-authors from Life Science College here at BYU. We developed this together. And the idea in biology is there's so many different experiments you can do, and there's so much field work that you could do. Biology is so much on field work that how do you do a virtual lab with that? So our approach here was diversity. You know, how do you look at all the species that exist in the world, and how do you organize them? How are they related? How are they different? How do you explore and measure and catalog those differences? So that's kind of the idea is you use these microscopy, and you use genetics and molecular biology and ecology as a way to explore those differences. And then you use this lab here, systematics, to organize these species and find out how they're related to each other. So it's all around diversity from different levels. So we have a microscopy lab, and each of the labs have these species selectors, all right? So what we've done is we've created a virtual world of these different kinds of species that represent all the different things going on in the world. So it's a small set, about 160 species. And you have different ways of organizing them. So all the species that start with B, for example, all the plants, or all the animals, or all the bacteria. And then you can organize them by characters. So all of the extinct species that we put in here. What about all of those lacking a nucleus? And, you know, or we can do it by classification. What's all the insects? What's all the amphibians? And so on, biomes and models. And so different ways of picking these species and say, okay, how are they all the same? How are they all different? So I'm going to come down here to paramecium because it's a nice one to show. So in the microscopy lab, we have all of these different microscopes. So we have a field scope, dissecting scope, compound scope, SEM, TEM. We also have clipboards here for different presets. And so each species has some set of pictures. So we have about 3,500 different images and videos of these different species. And then you pick a species and it picks the right microscope so you can see what information each scope gives you. So we have a chart that talks about magnification. So here's an example of, you know, some videos. So we took these pictures for the most part. We have some from public domain, but most of these are ours that we took. So here's the paramecium. So I think this is cool. And I'm a chemist, right? Look at it on a different scale, or, you know, look at what these paramecium are doing. And then we add staining. So here's some images with different kinds of stains and what you can see from these different stains. And then you can go to the SEM, and now you can look at that paramecium from a whole different world and perspective using this SEM image. And so this is what microscopy does. So you can look at all these images. We actually have animations for different things inside of here. So it's a really rich media environment. So it's, of course, a student can look at all those images anywhere on Google, but A, you're not going to find all the images we have, and B, it doesn't provide the context of where those images come from and what they're telling you. We have a genetics lab. So in the genetics lab, we have a smaller set of species, but what we've done is we've taken some traits. So for example, if I take fruit fly, and we have a list of traits, and we've created the genetic models for each of those traits. So some of these models are just simple Mendelian, some are sex-linked, some are incomplete dominance. We have linkages between, so if they pick two, you may see the linkage between two sets of traits. So we have epistasis, we have all of these different kind of models all built into these different species with these different traits. So what if I dig head shape, right? Which I think is leg-headed, you know, crazy thing. So what if I can pick the male and female and make it heterozygous, you know, or you can just have them pick them randomly, and then what you can do is do a cross, and you're going to see that they're going to get 100 offspring, and now you can see the statistics from the male and female, so you can see if there's any sex-linked genes going on, so you can see the parents, see the F1 offspring, and then you can also look at each of the offspring as individuals and look and see if you see different head shape. So you just kind of go through and see all of those different kinds of things, and you can do a next generation and pick parents from this generation and see how they go. We have them for pea plants. So we have all of these traits, seed color, pod color, humans, you know, we have real blood types. So we have A, B, and O, and then we have Bombay type that goes into it. Color blindness, for example, and so you do heterozygous there and do a cross. And we only get one cross because you only get one kid at a time in general. So you have to do a lot of crosses to start seeing what your statistics are and start seeing what's going on. So that's the genetics lab. So instead of just saying, here's some data, what you Punnett square, you know, have them generate this data, start analyzing it, and start seeing how well it agrees with Mendelian statistics. We also have population statistics we can do. So you can calculate gene frequency over time, and then you start changing parameters. Like what happens if I have a mutation rate of 0.004%, you know, calculate it, and then you can see your migration of your genes over time. Again, lab book, save this data, clipboards, presets, all the same thing. Molecular. So in the molecular, what we've done is we've taken every species in our virtual world here. We went to GenBank and we decided on 20 to 40 genes that we tried to keep common between different classes of species. So mammals have a set of genes, insects, and so plants. And so we have a mini genome that we've created with random sequences in between. And what you can do is take an epinorph tube, and then you can pick, like the mayfly, for example, and add DNA to your epinorph tube, and then you can add your nucleotides that are tagged and do another, add your tag. And now what you can do is then click on your primer refrigerator, and now you can pick your 2 primers so that you can take it to the PCR and amplify some gene that you're trying to isolate. Now I'm going to cheat and take my preset here. So I have fruit fly and housefly ND1 gene. So I've got it all. I got all the primers selected. Everything is set. You don't have to do that, but they're set. I'm going to turn on my controls. I have a little animation that plays here, and I'm going to turn on my PCR and just start my PCR process. So here's, it's going to be 1 minute at duration at 94 degrees at denaturing and so on. I'm going to come down here because this is going to take several hours and advance my time by a minute. So now I'm in the denature step. Now you can see animation so the students can see what's going on. Now I finished. So now I'm going to go almost done. Now I'm going to start going now to my next step, which is advance myself a minute. So I'm going to go to anneal. Then I go to, here's my annealing step, and then I'm going to go to the elongation step and show an animation. So I'm just going to advance several hours and wait till it says complete here. So it's complete. And now I'm done. So what did I get? Was I successful? You know, so now I'm going to go do a gel. So I'm going to take the fruit fly, add it to well 3. And I'm going to take my, housefly, well 6, turn on my power supply. There's my gel. So I preset these electrodes, but the electrodes can get switched if they do it wrong. I've set my voltage to whatever I want so they can melt the gel if it's too high. Duration you can change. This is enough. So I'm going to click on, and now my gel is working. So now I'm going to go advance this. So there's my tracking dyes. And I'm done. All right. And now I can turn on my UV light and say, okay, here's my standards are over here. I can click on my little measure thing and say, oh, that's about 1,077 base pairs. So that says, okay, I worked. I can save that to my lab book and I can shut it down. Now what I can do is take it to my sequencer and take one of these guys, drag it to my sequencer, close it, and now I can start the sequencing process. So I don't know how long it takes. So now you can see that I'm advancing my time. I can see my chromatogram as it comes off. And you can go do this and do a chromatogram here. I'm going to clear the lab and just start with something we've already done for the 2 genes. And so here's their sequences, which can be now, I can click on this, save it, click on this, sorry about that, save that, and then go to my lab book, and there is my fruit fly. So I didn't actually miss this guy, so we'll save that and go back to my lab book. And so there's my sequence for the fruit fly. For the other one, here's my gel. So, so that's how it kind of works here. So, so that's molecular. Lots of cool things you could do there. Ecology. I'm just going to go really quick on this guy. So here you can pick different biomes. And you can create your own biome, northern hemisphere, southern hemisphere. You can change your elevation, for example. And so this image kind of shows what happens when you get in the mountains, or I can go and change to like tundra, you know, and change what, and come over here and change what my mountains would be like in the tundra if I had those. Change your nutrient level. So there are all kind of these little variables. And what you do is you can pick species and throw them together in these biomes and then see what happens to their populations over time. So again, we got presets here. So here's a classic wolf and an elk, and I'm going to click start. And so I'm doing 1 year. So these units are in days. So it's 1 year is like every 30 seconds or something like this. So this is my population of my elk, population of the wolf. So we have these models created for all of these, and they interact, so you can see where their births are. You see that the elk are being eaten, their populations are going down. Some of the wolves die, but in general, they're going up because they have enough to eat. And then pretty soon, my elk crashed. Now my wolf crashed because they didn't have anything to eat. Now the elk are starting to go up because they're not being killed by the wolf and so on. So we have lots of these different kinds of invasive species. Now you can go get a cougar and a mouse and a red-eared slider, and then throw in a Burmese python and see what happens to those as you go and do those experiments. So systematics is the last one here in biology. The whole idea here is you pick these different types of taxonomy schemes. So Linnaean, a simple fake one, tree of life, that kind of the real one, and you can pick these different ones. And then you see all of these species, and you take a target species and you know to say, where does it fit? Where does this species fit? So I can take bacterium, and then what you can do is, for example, pick archaea, for example, and it shows you all these definitions of what should fit in archaea. So obviously, this is my discard pile, so I was wrong. So I'm going to come back here to the beginning, and it's a bacteria. And so you start picking these different kinds of species and organize them and see what's related to what. All right, questions about biology?
Go ahead and unmute if you've got questions on this. I haven't seen any in the feed on how biology works.
All right. Let's go do organic. Organic chemistry is divided into these 2 lab benches, synthesis and qualitative analysis. Organic chemistry tends to be a little bit more procedural than the other labs. They're still open-ended, but organic chemistry is more of these series of steps. I pick starting materials, I pick a solvent, I got to pick a reagent, I got to pick my reaction conditions, I got to do spectroscopy. So, it's really nice to have these worksheets to kind of go guide you through. So this first worksheet, it's like the first one anybody should use is just do thin layer chromatography, because it teaches them the process I'm going to show you. It steps them through each one of those steps and how to use the different tools. So this is kind of like a really good intro one to start with. So that's in the worksheets. I'm just going to show it to you without using the worksheet. So I'm going to go to synthesis first. So it's really busy, lots of things you can pick, but there's really not much you can do until you do certain processes. So it's not so bad. So what we do in synthesis is we have these 15 different named reactions. So if I pick Diels-Alder, for example, those structures are the starting materials that you are given if you pick this reaction. Or hydroboration, these are the alkenes you're given, or alkene hydration, or alkyl halide solvolysis, right? So these are just starting material sets. So it doesn't mean that you have to do esterification. It just means that these are the starting materials that are geared around an esterification reaction. So if I pick that, these become the starting materials that are available. So I can pick 0, none of them, 1 or 2 of these starting materials, and add it to this round bottom flask. And I don't care what combination or order that you do it in. And you can pick none or 1 of these solvents and add it to your flask. So for example, if I pick, I'm going to pick this acid. And if you want to know what it is, you mouse over, show what it is. Here's showing what's in the flask now. And then I can pick other acids. So I could pick another acid, even though it's not what I need. They could pick it and run a reaction and see what the real results are. So just because it's wrong, we don't prevent them from doing wrong things. So here's an alcohol. So I'm going to go add that. And I don't need a solvent. I'm not going to add it. So now I drag this down to my stir plate. So now my stockroom's closed. And so here is my flask. So I could click my stir button and start this reaction, but I'm not done yet. I can add any of these reagents I want. I can do it then, or I could go build my setup. So I'm going to go add heat. I want this to go faster, and I'm going to go add a condenser. And so, oops, I missed. I could double click, add it as well. I could go add my sulfuric acid here. And now I've added my acid. So a student may say, hey, I built it. I got my condenser. I got my heat. I got everything I need. I'm going to click start, and they go on their merry way, but they forgot to add the nitrogen, right? So it's a sealed system. So what would happen? Well, they'd think, okay, I'm going to go measure TLC. So here's my TLC. So here's my starting materials, and here's my starting material. Here's my reaction mixture lane. So here's my product. So I can save this, clicking save to the lab book, and then I could advance my time 10 minutes, for example, go do another TLC. Hey, my reaction's going, I'll save that. And then I'm gonna go advance another 10 minutes, and boom, it blows up because I had a sealed system, had it all clamped into place. So yeah, it's an exaggeration, but the kids like to have fun with the blow up. So you can find all the different ways we can blow things up. So let's do this for real and take it to the product. So I'm just going to go back and start my reaction. So I'm going to go here. I'm going to go add my heat, go add my condenser, go add my nitrogen. You can also use ice. So we have all these reactions that they run on ice. You can run it at room temperature. We have kinetics running for each of these. So we know the kinetics, first or second order kinetics, and we know how long they take. We know the rate constants. So all that's in this in the simulation. So if they use ice, then it's going to be really slow, or maybe they have to use ice for certain reactions to slow it down. So same thing, TLC. There it is. Now I'm not going to get products because I forgot my nitrogen, so I'm going to start over again. So I'm not doing very well. I can double click on these guys to go at it. Let's just go do it quickly. Here's my sulfuric acid, double click, double click, double click, double click on that. So now I've added it. So you can drag or drop or double click, have everything I need. Now I'm going to start. So we're going to go advance my time. There's my product. Yes, they can see that their product's being made, but what they don't know is how long. So what we do is we grade them on how long and can they report the length of the reaction from that, the kinetics of it. So here's I missed it. Here's the TLC. So we're almost done. So I'll just do 10 more minutes and prove that I'm done. Save that, say okay. So now I'm ready to work it up. So drag the sub funnel. So now I have my reaction, my product mixtures here. And now I can add any of these aqueous reagents to work it up. So you can do countercurrent extraction. So you can add a base to go pull off your proton to maybe go into the aqueous layer to put something else into the organic layer. I'm just going to go add nitrogen here. And there's my organic. Now I'm going to go, by picking my organic layer, I'm going through a rotovap step. I could also, if I wanted to, if I had something interesting in my aqueous layer, I could pull it off and have my two different layers separated in the flask. So now, how do I know it is what I want? So now you can do your spectroscopy. So here's proton NMR. And so there's your peak. So each of these peaks are numbered. And here's the integrations down here at the bottom. They're random. They're all consistently integrated, but we choose random numbers each time. You can zoom in to see what the splittings are. Save that to the lab book. I can do a carbon-13 by clicking here and changing the probe to be carbon-13. And now I can see my carbon-13 peaks. Now I can do FTIR. And these are all real spectra that we acquired from Sigma. So save that to the lab book. And we can do mass spec. So there's your mass specs. And you can still do a TLC on this, so you can measure what its polarity is. You can also do melting points based on if it's a solid, and you can do distillations if you need to do a separation. So this is kind of how the synthesis works. And we've got about, I don't remember exactly, 750, 800 different spectra. And then here's where they're all saved in the lab book. And so lots of things you could do here, all of these different reactions. So we have, I think, about 5 million different permutations of what you can do. Many of them don't work, don't give you any reaction, but we let them go do that. So any questions on synthesis before I go to qual?
There were a couple that I wasn't sure on. Can you add multiple two acids and an alcohol in this specific reaction? So how many options on what people can add in are there for these reactions? I didn't know.
So for starting materials, you can add any combination of 0, 1, or 2 items. So you can add none, 1 item, or 2 items. So you can add 2 acids, 2 alcohols, whatever you want, and we'll have that outcome. Anything else?
Other question was, says I'm still muted. Can you hear me?
I can hear you.
Okay, great. Can we make the text larger in the integrations for the proton NMR? So can that be zoomed? It can all be zoomed, right?
So when you save it to the lab book, it's bigger. So like here in the lab book, it's saved in a larger format. But no, this was developed back in the days before we had, the monitors were smaller, and 600 by 400, and we were using fixed graphics. So we do know the labs work on to accommodate various needs. Like we have screen read, our worksheets work with screen readers and screen magnifiers. You can use a screen magnifier to get bigger if you need to.
And you can also just like pinch zoom the screen and it does make it bigger, or just doing a display zoom. So it's not easy, but yeah, you can zoom up on them.
All right. Coming up, almost done. So qualitative analysis. So the idea here is teach students how to analyze their compound so they know what it is, right? And so what we do here on the chalkboard is we have these different functional groups, and each of these functional groups have 2 or 3 or 4 different knowns that they can practice with to interpret spectra and do wet chemical tests. So those structures you see on the knowns, not the unknowns, they're just knowns. And so they can pick one of these, like an aldehyde, and we also have a general unknown. So it could be anything in our database. And so, so if they pick an aldehyde, what you get here is you'll get these 3 different knowns that they can practice with, or they'll get an unknown. And the unknown is numbered, like number 210. So again, we have an instructor manual, answers to all the worksheets, and we also have answers to what the unknowns are. I believe there's like 250 or so unknowns from which it randomly generates. So each student will get a different unknown. And so what you can do is pick that unknown, for example, and add it to the round bottom flask. And now you put it on the cork ring, and we give you unknown number, boiling point, percent carbon, percent hydrogen. And so now you can do these different tests. So again, go do NMR, right? There's the NMR of this particular compound. This thing is you can zoom in here, you just can't zoom in on the numbers. Then you can say do the FTIR, and so you can do all your spectra that you want. And now you can do these different wet tests, right? Wet chemical tests. So I take an aliquot, put it in my clamp, and I'm going to show you a real picture of what that unknown looks like. And then we can do any of these tests. So Jones oxidation. And so we're going to play a video of what the result is for that test, or we're going to show you a real image of what the result is for that test. And so this is positive, and so it gives you a positive test, so they can go through and do all of these different tests, iodide test, and acid test, and Hindenburg test, and so on. And so they can go through, and then they would just report their unknown to you and say, hey, I got an unknown 210, here's what I think it is. So that's one way of doing it. Another way is to go to the clipboard again, and you have these random unknowns, same idea, but I can go pick like any of these unknowns, so like general unknown, and it's going to, somehow I missed that. Let's just do aldehyde. And so it's going to give me an unknown, but there's no number. And so I can do my spectra, do the same thing. And then when they're all done, they'll go to the lab book. Here's the report button. They'll write it in, my unknown, and then they'll click submit. And in this case, we show the answer to them. Now you think they can cheat. Well, they really can't. They can cheat, but it's going to be more work to cheat than it is to do the right answer, because if they go back and they want to get another, if they get another unknown, we give them a new random unknown. So they'd have to go through 20 or so unknowns to go get the same thing again, and then they wouldn't know, right? Because it all looks like a clear liquid. So, you know, it is possible to cheat, but the number of ones they cheat because over time the students will get to know, oh, 210 is this. And so just this is, I'm not saying that either way is ideal, but these are two ways of doing unknowns. Then you would just save the file, right? And save that, and then then the student would upload that to the LMS, and a TA would open these files and look at their results and grade them based on that, on what they've done. So that's kind of how we do it. Any questions about qual?
Hello, Professor Woodfield. I had a quick question for you.
Uh-huh.
Can you imagine using these organic synthesis or qual analysis labs for, say, an honors course that we have to teach like online for the lab? At the college level.
At the college level?
Yeah.
Yes, absolutely.
Okay.
Because,
Because we have this quarter, for instance, I'm from Dartmouth, and we have to teach an honors organic lab. And in the normal lab, we have them do like a multi-step synthesis for over 9 weeks. So we're trying to come up with like some replacement for that. So I was wondering, are there like, probably in synthesis, yesterday I was trying out some aldol reactions, and there's one reaction with an ester where stereochemistry plays a major role, you know, in deciding the outcome of the aldol reaction. So I was wondering, based on, you know, your experience, are there like particular experiments that you could suggest that would be suitable for an honors course?
So that's a great question. So the way the simulation's configured is we don't allow we don't allow multi-step reactions, because the permutations quickly got out of hand and we wouldn't have been able to finish the project. But what you can do is you can use one reaction to make like a product, and then you can take a same or similar starting material from another reaction and start that. But I'm not an organic chemist. I'm a materials chemist, solid state physics guy. And so the organic lab was developed with my colleague, Merritt Andrus. And what I suggest you do is you email me those questions, and I can put you in contact with Merritt. And he's the one that designed all of the starting materials and the reactions we did. And he would definitely be able to talk your talk and help you out.
Okay, great. Thank you so much.
Brian, a bunch of people have asked, and I don't know the current status. Can we integrate Beyond Labs with Canvas?
So here's what we can do for we can do with the labs. The labs are installed on local machines, so Mac and Windows 10. Okay, we are working on HTML5 browser-based versions, but those are a year for, probably do chemistry first, and that's a year out. And that doesn't help us in the current situation. So we are trying to get to the web delivered version, obviously. But what we can do, and we know it works because we have several customers doing, is we can integrate the download and the assignment of the license from within Canvas. So if, that's a capital I, capital F, if you are having your students, purchase their own codes and the school isn't doing it, then what we do is we can integrate that license purchase and assignment part into the LMS as a LTI integration. So students just have to go to one location to go download the product and then to go get their license. They get the license sent to them, activate it, and they're done. If you are going to purchase the license for the students, as part of the lab fee or whatever it may be, Then what we do is, the easiest thing to do is to tell us how many codes you need, and we will create a single code for your school with the X number of uses or installations put into it, plus some free ones for you and your faculty colleagues or TAs, and then you would just send that to the student as a single code, and then in the LMS you would just put the download site inside the LMS. We can give you the download site for the app. As for the PDF files, they're just PDF files. So the best thing to do is to just pick the ones you want, download them, and load them into your LMS system as PDFs. Now, so I'm going to anticipate a question that was happening in the last webinar, is what about the PDFs? Yes, we would love to integrate those into HTML5 docs, and that's on our development plan. But again, for the current situation, they're not available. It was great to do PDF response forms, but that actually turned out to be harder than it was to make an HTML file. So in the interim for these PDFs, here are what people are doing that are using it. The easiest way is, say, have the student print out a PDF, fill it out by hand, and take a picture of their results, make a PDF, and upload it into your LMS. An alternate solution is a student creates some type of document, like a Word doc, whatever, and all they do is put in their answers and screen capture different parts of the lab. And so they're kind of creating a lab book kind of thing of just that matches to the worksheet, but the worksheet's not in it. They just put in their answers, screen capture their spectra or whatever it may be, answer their questions, and upload that into the LMS. That's a really easy way of doing it. Sometimes some of them are asking them, like I know when I'm a Mac user, in Preview, you can pretty much add text anywhere you want. So that you can take that PDF, add the text, and add graphics to it and save that electronically and make it all electronic as well. So those are pretty much how people are doing it right now. But of course, you don't have to use our lab activities or worksheets. You're welcome to write your own or modify what we have. And if you want our Word versions of them, we can give those to you. All right, any other questions?
Yeah, again, from Pushpa at Dartmouth. Would some of the general chemistry experiments, particularly quantum chemistry experiments, be useful, like for upper division PChem labs?
Absolutely. So Western Governors University, for example, uses our general chemistry calorimetry and quantum labs for those upper division PChem courses for what they're teaching online. Absolutely. And we do have physics, but that's not really relevant for chemistry, but we do have physics labs with some optics and circuits and mechanics in it as well.
Okay, thank you.
So Brian, how do we compare to Labster?
Okay. I would say, in my honest opinion, you know, since late night labs has disappeared because they're no longer working, you know, there's PhET, there's us, and there's Labster. You know, and you know, from a teacher standpoint, from a educator, you know, they all do things differently and hit different parts of what you're trying to teach in science. So PhET is really a small, well-controlled, what I would call single variable experiments. So they're great, but the only control, essentially, sometimes two, but essentially it's a 2D interface, and it's all about what happens as I change this one variable, and it's really focused on those things. Labster, their focus more is on that interface and creating a VR type real-time rendered interface. But if you really go to their simulation engines, they're what I call, they're on rails. So it's like the old Journey video games from 20 years ago where you went down on a journey and showed you some cool pictures, and maybe you could take one step off the path or two steps off the path, but you always had to get back on the path. And that's what Labster does, is it's really geared toward that type of approach of a story. So they have their activities are story based, and they're good. They're interesting, and it's engaging to have those graphics, but you don't get the options. They're not open-ended, right? You don't get 12 different or actually 16 different acids and bases that let you do polyprotic acids and tribasic bases with 8 different indicators and all those kind of things. And so that's a different approach. Our philosophical approach is open-endedness. I invented the idea as a grad student at Berkeley years and years ago. And the whole idea was, as a TA teaching Chem 1A at Berkeley, was these students were cookbooking, and I wanted to get them away from that cookbooking. And so the only way that you could safely and economically do that was to create a virtual lab. And so that's the idea, is to open-ended, environments where students have to make decisions and figure out why things don't work or why the results aren't what they expect. And I think that's really what the key is to being a scientist.
Any other questions? I think we're good for tonight, Brian.
All right, thank you for coming, everyone.
Professor Woodfield, I have one last question. May I?
Of course, yes.
Okay.
Oh,
Right. With the organic synthesis, one thing I noticed when I tried some of the labs was there's no stoichiometry at all with the synthesis. You just add the reagents, but with no respect to molar ratios or anything. Is there something, could something be done to introduce that concept too? Because I think that's very useful for students.
So, great question. And yeah, I think that the way that we've developed the database, the relational databases that drive that simulation, is that could be done. Really, it's a matter of what we had to decide was, we had to decide, here's our budget, here's our time, here's the resources we had available, what was the best thing that they could do. So it was around the, really we designed these to be augmentations for the labs. I'm a chemist. I'm not going to I want my students to be in real labs, but here's an anecdotal story where the ideas came from. Was, my dad was the chief software engineer for Apollo, and my dinner table conversation was how to write code when I grew up as a kid. And his mantra was always, a computer is only a tool, and it's good for some things and it's not good for other things. And so simulations aren't good for teaching those kinematic skills for a real lab and some of those quantitative things. And so what we decided to do was to implement what a computer and a simulation would be good for, is decision making. And so for organic, it's decision making of what reagents under what conditions, and not the quantitative side. But we do have other experiments like titrations, it's quantitative, calorimetry is quantitative, and so on. So you're right, we could do that. We just haven't had the bandwidth to go add that yet.
Okay, thanks.
All right, I think we're good. I appreciate everyone coming, and I hope this was helpful. And if you have questions, just use the contact page on our website, or even better is to ask questions on the community page, because then as we answer those questions, others can see the answers and get more information more easily than having to wait for me to respond to email. So we appreciate your interest and hope we can help you at this time. We're all crazy. It's all crazy stuff, so we'll help you the best we can. All right, so we're gonna end this.
Thank
Have
you.
a good night, all.
Thank you everybody. Thank you for coming.